Hello, everyone, and welcome to TriLink's first ever Spotlight workshop. My name is Taylor McReynolds. I will be your moderator today. And today's workshop will be on CleanCap co-transcriptional mRNA capping. We'll be having a Q&A session at the end of the presentation, so please submit any questions you may have. At this time, I'd like to introduce our presenter and TriLink Senior Director of Emerging Sciences and Innovation, Dr. Anton McCaffrey.
Hi, thanks everybody. So welcome to our webinar. So today I'm going to be telling you about our co-transcriptional cap analog, CleanCap. But first I'd like to tell you a little bit about what TriLink does. So we've been around for about 23 years and we specialize in highly modified oligonucleotides and nucleic acids. These include single guide RNAs for CRISPR research and other highly modified oligonucleotides. We also do custom NTP synthesis for a variety of applications, including diagnostics and clinical applications. Today, we're going to focus on messenger RNA, and we are the leading manufacturer of custom messenger RNAs in the research and clinical spaces. And today, we're going to focus on a particular aspect of that called CleanCap, which is a co-transcriptional cap analog that is important for manufacturing messenger RNAs for a variety of applications. And lastly, we're the leading GMP manufacturer for nucleic acids, including CleanCap, messenger RNA, and synthetic guide RNAs. So, why should we be interested in messenger RNAs? So messenger RNAs are useful for a variety of different applications, including genome editing using CRISPR-Cas9, zinc finger nucleases, and TALENs. mRNAs are also useful for gene replacement, and they're also interesting for a variety of immunobiology applications, including the creation of engineered CAR T cells, vaccines for infectious disease, and for personalized cancer vaccines, and for self-replicating RNAs such as alpha viruses that are used in vaccines as well. So the molecule that we'd like to make is a messenger RNA. And so just let me walk you through a little bit about what the different aspects of the anatomy of messenger RNA. So at the 5' end of the messenger RNA, we find a cap. And this is going to be the focus of today's talk. This is a 5' cap that's involved in initiating translation. So it binds initiation factors that recruit the ribosome to come to this messenger RNA and start translating it. It has a 5' untranslated region, and then a coding sequence, which is determined by the customer. Basically, this is the thing that's going to be expressed by the ribosome when you put this messenger RNA into the cell. Then there's a 3' untranslated region and a poly A tail. The poly A tail is important for bringing together the cap and the 3' end of the messenger RNA in order to enhance translation, and the ribosome goes around in a circle, making many copies of protein from this messenger RNA. So what are the functions of the messenger RNA cap? So it's involved in nuclear export, in splicing. It's involved in translational initiation, as I just mentioned. It also regulates decapping and turnover of the messenger RNA. And another important aspect of it is it determines the self-non-self recognition of a messenger RNA. So your cells have pathways that recognize whether a messenger RNA belongs to the cell or perhaps could be from a virus. So there's these pattern recognition receptors within your cell. They function within the endosome. And so, for example, toll-like receptors recognizing single and double-stranded RNA. And then there's a variety of cytosolic receptors that recognize improperly capped RNAs, which will be a focus of our talk today. These include IFITs and a protein called RIG-I. They also recognize double-stranded RNA, so these are PKR and MDA5, and also DNA ends through the seq-fas sting pathway. And these different pattern recognition receptors are there to recognize RNAs that shouldn't be there, for example, from a pathogen such as a virus. So this is what the 5' end of a messenger RNA looks like, except this one is not fully formed. So this is called a cap0 structure. And in the cap0 structure, the 2' position of the first sugar contains an OH. And this is not found in higher eukaryotic messenger RNAs. Indeed, most, if not all, messenger RNAs have a 2' O-methyl group at this first sugar position. And this structure is called CAP1. And this will be important in this talk. There's also another CAP structure in which there's a second methylation event at the N6 methyl position of adenine. And this is known by biologists as M6AM. So it's a CAP1 structure, but also has an additional methylation group there. And this is thought to, in some fashions, control stability of the messenger RNA. So why do we think that CAP1 is important? So CAP0 is recognized as foreign. And these proteins that I alluded to earlier, IFITs, they recognize non-methylated caps and sequester them from the translation machinery. So CAP1 methylation reduces binding to these pattern recognition receptors that are looking for foreign RNAs within the cell. And we know this is important because many cytoplasmic RNA viruses employ a CAP1 methyltransferase. And when you block that methylation event, what it leads to is a virus that's attenuated. There's also another structure called CAP2, which is largely unexplored until this point. So how do we go about putting a cap on a messenger RNA? There's really two different buckets here. So one bucket is enzymatic capping, in which we use a set of proteins from the vaccinia virus that do capping and methylation at that first position. However, these enzymes are expensive at scale, and they can vary in their efficiency of capping. So people developed another way of doing this, and that is to do co-transcriptional capping, in which we include a cap analog in the messenger RNA transcription reaction. And that cap analog, it caps as the RNA is being transcribed. Now, there were a first generation of cap analog called anti-reverse cap analog, or ARCA. But today I'm going to tell you about a revolutionary new method for capping RNA co-transcriptionally that we call CleanCap. But first, let me tell you how ARCA works. So this is what the molecule called ARCA is. It's a dinucleotide cap analog. And this cap analog can sit down at the first +1 position of the promoter and initiate transcription and add a cap. However, during that process, GTP, which is in the reaction to extend the transcription, can also initiate at this +1 position. So you have a competition between these two different cap analogs and the NTP. And in order to do efficient capping, what's normally done is you put in a large excess of this cap analog so that you get more capping with the cap analog and less with the GTP. However, this leads to a reduction of yield because you're starving the reaction for GTP. And so we came up at TriLink with this new co-transcriptional cap analog that we call CleanCap. Instead of being a dinucleotide, it's now a trinucleotide. And here you can see the cap analog sitting down at the +1 and +2 position of the promoter and initiating transcription. And because the affinity of that interaction is higher than with the NTP, and because we've chosen an NTP at the +1 position, which is not desirable for T7 transcription, we can use actually an excess of GTP over the cap analog and still get efficient capping. The result of this is that we get about 2.5 times the yield of the ARCA reaction. And in fact, when we look at the capping, we get about 3 times as much capped RNA from a given amount of a T7 transcription reaction. So at TriLink, we also developed a capping assay in which allowed us to look at the identity of the cap of the messenger RNA. This is challenging because we're looking for the addition of a single methylation event on a very, very long RNA. And so in order to achieve this, what we do is that we chop off the end of the RNA, and then we analyze the fragment on the 5' end through LCMS. On the left-hand side of the middle part, you can see a capping reaction with an ARCA capped RNA, in which you have the ARCA capped RNA, but then a significant amount of uncapped RNA. In the middle, you can see an enzymatic capping reaction that hasn't gone to completion, and you can see the various partially methylated events that have happened in there. And then on the right, you can see a capping reaction with CleanCap, where we can see that the majority of the mRNA is capped, with the small amount being uncapped. So why is this important? So experiments in mouse liver showed that when you have a cap 0 RNA, that this is capped inefficiently and gives a cap 0 structure. This is recognized as immunogenic, and the cell shuts off translation of this RNA, and you see expression at 24 hours that's similar to the PBS control. Whereas with the cap 1 RNA, which has an authentic cap, this is expressed efficiently within the cell. One other approach that we use here at TriLink is that there are some things called self-amplifying RNAs that are interesting for vaccines. And these self-amplifying RNAs are based on alpha virus backbones, and they replicate once inside the cell. And so these are interesting for vaccines because during that replication process, this looks like a viral infection to the cell. And as a result, you develop good immunity to any antigen that is expressed from the self-amplifying RNA. And CleanCap AU streamlines the production of these self-amplifying RNAs, or saRNAs, for vaccine production. It reduces the number of manufacturing steps, it reduces the number of proteins required, and reduces the cost. And all of this accelerates vaccine manufacturing. And on June 19th at 9 o'clock PST and 11 a.m. Eastern Standard Time, we'll have a webinar focusing on CleanCap for this particular application of self-amplifying RNAs for vaccines, which is quite timely now in the age of COVID. Lastly, I'd like to focus a little bit on GMP manufacturing at TriLink. So TriLink was the first contract GMP manufacturer of messenger RNA in the world and still is the leading manufacturer. So we have a well-qualified quality system. We're ISO certified and fully compliant with ICH guidelines. We have a fully traceable document system. We're frequently audited, and we support a variety of different applications, including basic research, diagnostic GMP, preclinical IND and toxicology studies, and phase 1 clinical trials. So at TriLink, we have over 24 years of nucleic acid manufacturing experience, and so we're well seasoned in this area, and we bring more experience to this than any other contract manufacturer in the field. We pioneered the manufacturing of messenger RNA for contract manufacturing in this space and have been doing this for longer than anyone else. We have a management team that's proven in the life science industry with leaders with extensive operating experience. We have greater than 200 employees, and we're growing, and with 1,000 years of combined nucleic acid chemistry and GMP program management experience. We recently moved into a beautiful new facility that was purpose-built for the manufacturing of messenger RNAs and small molecule critical components such as CleanCap. So we have 5 new ISO Class 7 and ISO Class 8 rooms that are engineered for GMP manufacturing in our custom suites that were purpose-built for just this application. So to conclude today, CleanCap co-transcriptional capping generates a cap 1 and M6A mCAPs that are efficient. It gives more efficient capping than with anti-reverse cap analog or ARCA. CleanCap yields 2.5 times as much messenger RNA as ARCA, and it's cost-effective and more cost-effective than enzymatic capping. And as I mentioned before, when you do capping with ARCA, you achieve what's called a cap 0 structure. And this is expressed poorly in vivo. So as I showed you earlier, similar to the PBS control, whereas a cap 1 structure is efficiently translated by the cell because it's recognized by the cell as a self-RNA, and that it allows it to be translated efficiently without inducing these pattern recognition receptors that can inhibit translation. And I alluded a little bit to CleanCap AU, which is going to be the subject of our next webinar. So CleanCap AU provides the 5' end that's authentic for a self-amplifying RNA, and it accelerates the production relative to production with enzymatic capping by reducing the number of steps that are involved in this process and reducing the number of proteins that are involved in the manufacturing process. And as I mentioned, TriLink is a leading contract manufacturer for GMP grade mRNAs and vaccines. So when you're ready for your project, come talk to us. So we have a team of scientists, including Taylor, who is your host here today, that can guide you through the process of designing and implementing your messenger RNA program, even without having to know a lot about messenger RNAs. So we can guide you through this process, educate you about what you need to know and how you can convert your application to a messenger RNA kind of application. And our product management team, manufacturing, quality assurance teams can work together to deliver to you the best possible solutions for your messenger RNA applications. So if you're interested in learning more about this, you can contact myself at this email address, or you can contact our support teams at TriLink, and they can guide you through this process. And so with that, I'll conclude, and I'll turn it over to Taylor to see what kind of questions you guys might have for us. So Taylor, do you have any questions for us? Are you on mute?
Sorry about that. I was on mute. But yeah, thanks for that wonderful presentation, Anton. Let's get right into some of the questions. And you guys can still submit your questions if you have any. So here's a good one. What is TriLink's IP position on CleanCap, and are other organizations able to make this analog?
So CleanCap is a patented technology that belongs to TriLink, and so it's something that you cannot get at other places other than TriLink. So we developed this technology, and the IP belongs to us. However, we've made this available widely throughout the community. And so basically our philosophy is that we would like to enable as many people as possible to incorporate CleanCap into their research programs and into their clinical programs. And so it's something that's proprietary to TriLink, but also freely available. So we're basically Switzerland. We'd like to work with everybody. And so you can come to us and use this technology for your manufacturing.
All right, and then here's another one. Are you able to give some just general cost comparisons between the three methods of capping that you mentioned? So ARCA, CleanCap, and enzymatic capping.
Sure. So this relates to two different aspects. So there's the cost of the materials involved. So the two enzymes that are involved in enzymatic capping are difficult to manufacture. They're multi-subunit proteins, and these enzymes are expensive. And as you go to larger scales, you have to ask yourself the question of, can I obtain these enzymes in sufficient scales to make large amounts of messenger RNA? With the co-transcriptional capping with the anti-reverse cap analog, or ARCA, as I mentioned, you have to limit the amount of GTP in the reaction in order for the capping to be efficient. And as a result, the yield of the RNA is about 4 times lower than what you would get when using enzymatic capping or capping with CleanCap. And as a result, the cost of doing that is higher because you get a lower yield per milliliter of transcription. So in the end, CleanCap is something like twice as efficient and half as costly as using other technologies such as co-transcriptional capping or capping with the cap analog.
All right, and there's another one that kind of follows from that. So other than upfront cost for the enzymatic capping, is there other advantages of doing CleanCap to enzymatic capping?
Yes, absolutely. So especially when you're in the GMP manufacturing space, you want to simplify the process as much as possible. And so with enzymatic capping, generally you're doing a transcription of an uncapped RNA. You're purifying that RNA, and then you're capping it with enzymes. And then you're purifying that RNA again. With CleanCap, you can do a one-pot reaction in which you make the RNA, you purify it, and then you're ready to go. And when you're doing GMP manufacturing, each step is very carefully controlled and all the reagents are very carefully controlled. And so with CleanCap, instead of using 4 enzymes for your manufacturing process, you're using 2. And you're reducing the number of steps and number of purifications and doing this in a one-pot reaction. And the end result is that you have fewer enzymes to qualify and manage, and you have less steps that need to be regulated. And so this is a much more efficient process, and that greatly reduces the costs of GMP manufacturing by streamlining that process.
All right, thank you very much. Another question is, is there in vivo data comparing CAP0 to CAP1? This might be a question better answered offline, but I don't know if you have anything to mention about that.
Yeah, so I actually showed you some data. So this is data with the LNP formulated RNA that was injected by tail vein into the mouse. And this is expression in the liver of a luciferase RNA. And at 6 hours, which I'm not showing, you get some expression of the CAP0 RNA, but higher expression of the CAP1 RNA. By 24 hours, however, the expression of the CAP0 RNA is similar to the PBS control. What's happening there is that the cell is recognizing that there's a foreign RNA in the cell, and it shuts off the translation machinery in order to prevent that virus from replicating. And so you get very poor expression from a CAP0 RNA, whereas a CAP1 RNA looks like a fully licensed self-RNA, messenger RNA. And so the translational machinery recognizes it and translates it, and the pattern recognition receptors that I alluded to earlier are not recognizing this as foreign. And so this RNA is allowed to be expressed by the ribosome. And so CAP1 gives superior expression to CAP0 RNAs, at least in mouse liver in vivo.
All right, thank you very much. I guess there's 2 questions that kind of go hand in hand. I think you kind of touched on this, but does CleanCap change the purification methodologies, and what method does TriLink use or recommend? And then also, what purification strategy does TriLink use for GMP manufacturing?
So yes, so as I mentioned earlier, when you're doing enzymatic capping, you're generally taking the uncapped RNA and purifying it, and then doing the capping reaction on that RNA to cap it, and then purifying it again. There are a variety of different purification methods that are available for messenger RNA, and it turns out that this is actually the most challenging part of doing messenger RNA manufacturing. And so at TriLink, we have a variety of different purification methods that are suitable for different applications. So if you're going to do genome editing with messenger RNAs, for example, or protein replacement, then you want an RNA that has a longer expression and is not shut off in the cell. And for that, we use reverse phase RNA purification for messenger RNAs. However, for other applications, such as vaccines, the purification can be different. So in the case of a vaccine, and in our next webinar on CleanCap AU, we're going to get into this. impurities in the RNA, such as double-stranded RNA that would be removed during a reverse phase purification, can actually serve as an adjuvant for the vaccine. And for this particular application, we can do a less stringent purification of the messenger RNA. And for that, we've developed a method that we call LC isolation. LC isolation is good at removing process impurities such as NTPs and double-stranded DNA, proteins, buffers, those kinds of things, and giving you a messenger RNA that is relatively pure, but still has some double-stranded RNA for the manufacturing process. And this double-stranded RNA actually serves as an active ingredient in this vaccine and serves as an adjuvant. So TriLink has a variety of different purification methods that are tailored to your particular specific application.
Okay. There's another question here. I know you touched on this, but I think it's maybe useful to reiterate. Someone asked, how do you attach the CleanCap to the oligo? And I think that's one of the unique features of CleanCap. That.
Sure. Yeah. So here in the bottom, you can see the CleanCap analog that is binding to the +1 and +2 position of the promoter. So the promoter is shown in the blue region, and CleanCap is binding to the 5' end of the messenger RNA. And then hanging off the end is this M7G cap. And so once this CleanCap analog binds, you'll have the addition of the first NTP, and it basically elongates this oligonucleotide primer, which is this trimer, into a tetramer or pentamer, and extends this RNA. And so the capping happens co-transcriptionally at the initiation event at the T7 promoter.
All right, thank you very much, Anton. Another thing I think worth mentioning, does TriLink incorporate modified nucleotides into synthetic mRNA to increase stability?
Sure. So we didn't really touch on this in this talk today, but another factor that authenticates a messenger RNA as being a self-RNA is the presence of a small number of chemically modified nucleotides in the RNA. Now in the cell, the RNA is made and then those modified nucleotides are added co-transcriptionally. However, in a transcription reaction, we can also include modified nucleotides in the as NTPs, and these become incorporated in the RNA as the chain grows. And we found through extensive research at TriLink and with our collaborators that for applications like protein replacement and genome editing, that there may be significant advantages from incorporating modified nucleotides within the messenger RNA in order to reduce binding to these pattern recognition receptors, specifically the ones that recognize double-stranded RNA, such as PKR and MDA5. And so TriLink is a leading manufacturer of modified NTPs. We offer a wide variety of them in the research space, and we can also produce them in GMP scales. And so, yeah, it's for some applications you want modified NTPs. For applications such as a vaccine, for example, you may want to use a wild type unmodified RNA, which will be more immunogenic.
All right, thank you very much. And I guess going off of that, another one is how much does using 5-methoxy-U increase expression versus wild type UTP?
Sure. So through a variety of experiments and extensive experimentation really over a number of years, we identified a small number of modified nucleotides that seem to reduce binding and activation of these pattern recognition receptors. One of those that we identified was an analog called 5-methoxyuridine. And what we found was that inclusion of 5-methoxyuridine in messenger RNAs increased the activity in a variety of different cell and in vivo settings. However, what we also noted was that in order to fully use this 5-methoxyuridine, we also found that it was beneficial to reduce the number of total uridines within the transcript. And we can do that by doing silent mutations of a codon basically using synonymous codons that have fewer uridines in them, but still give the same amino acid. And so the combination of 5-methoxyuridine in combination with uridine depletion turned out to be useful for increasing the expression of messenger RNAs in a variety of different experimental settings.
All right, thank you. And I guess I'll try to do one more question. We're kind of at our time limit, but so for mRNA transcription using CleanCap, what enzymes are required?
So the enzyme that's absolutely required is T7 RNA polymerase, or you could use other RNA polymerase, but that's the enzyme that's fully required. We also include an inorganic pyrophosphatase. So what that's doing is breaking down inorganic pyrophosphate that's generated during the reaction. That is an inhibitor of the reaction. And so we include this inorganic pyrophosphatase to increase the yield of the RNA. And then often people will include an RNase inhibitor in the reaction. This is not strictly required if you have great RNase-free technique, but it's sort of a little bit of a seatbelt to ensure that if a small amount of a nuclease is present or introduced as a contaminant during the reaction, that you don't degrade your RNA. And then typically you do a digestion of the DNA template afterwards with DNase 1. And so those are the enzymes that are commonly used in the in vitro transcription reaction.
All right. Well, it looks like we're at our time limit. Thanks again, Anton, for the wonderful presentation, and thank you to all of our attendees. Apologies if we did not get to your question. If we did not, we will reach out to you via email. If you have any remaining questions, you can reach out to sales@trilinkbiotech.com. And I hope you can attend one of our other upcoming webinars, like the one Anton mentioned on CleanCap AU. Please see the events page on our website for more details. Thank you and have a wonderful rest of your day.
Thanks, everybody.